The last video was critical. We are learning to name structures that have more than one sugar in them, and in particular to talk about the linkage. Remember, a linkage between two sugars is defined by the numbers that are connected, which carbons are connected, and the anomeric state of any anomeric carbons that are involved with that linkage. That's why this molecule here, sucrose, was connected by an alpha-beta 1,2 linkage. By the way, we'll just go ahead and cross this one out. It's actually a slightly different form of sucrose. Let's not worry about it. What I'd like to do at this point is move on to the next page and talk about polysaccharides. And the good news is that once we've learned how to name linkages between two sugars, there's really nothing more to learn. We just keep adding sugars. In fact, most polysaccharides are quite a bit simpler than what we just observed. Let's give you some examples and we'll try to test you a little bit to see how you're doing. Here's an interesting molecule that we're going to talk about later in this class. It's called glycogen. And let's just make a few notes about it. It's found in your muscles, and it's found in your liver. And we'll talk later about why those tissues have it, but the upshot is that it's the short-term storage of glucose. In the human body. So if you have glucose extra from your diet and you want to put it away for later, meaning on the sort of the daily feast-fast cycle, you might put it in your liver in the form of this molecule. Glycogen is all glucose. There's no mannose, no galactose, no ribose, no anything, no fructose. It's all glucose, but it's linked together with two different forms, and it's branched, as you can see. That's quite different than proteins, which we noted were not branched. What we have is a bunch of glucoses. Let's zoom in a little bit here. We have a bunch of glucoses linked together in a straight line, and there's lots of these fibers that are going along, and those all have a particular type of linkage. Let's make a note that all of the linkages are the same in the linear portion. And then we have connections between these linear portions that we would call that the branch points. Okay, let's pause the video, and I'd like you to identify or name the linkages between the glucose subunits here in the linear portion as well as the branch portion. Pause and name those two linkages. Okay, so the first thing we got to recognize is that these are being connected between two carbons, and we have to come up with the numbers for those carbons. You can count them out, and it's 1 and 4. So this linkage is a 1 to 4 linkage, just like we saw for lactose on the previous page. Now, the next part is the tricky part that messes people up. The rule is if you have an anomeric carbon and it's alcohol, if that alcohol is buried in that linkage, you have to make a note of what its anomeric state is. So let's consider where are the anomeric carbons. The anomeric carbons are always number 1 in glucoses. So in this case, the oxygen that's contributed from the 1 position of this sugar is anomeric. The one that's contributed from the 4 position on the next glucose is not anomeric. Therefore, we only need a single Greek letter, anomeric indicator. This is down on that 1 position. This is up. That's opposite. That makes alpha. This is an alpha 1,4 linkage. What did you find over here for the branch? Well, it turns out it is also an alpha linkage from this 1 position, but instead of being to the 4 position, which is down here, it's to the 6 position. So the branches of glycogen are alpha 1,6 branches. That's how glycogen always is. So the last thing we'll say about glycogen is that we have lots of termini. there are branches, and that makes, like a tree, you would have a trunk, which is kind of over here, and a bunch of branches, which are out here. Now, here's an interesting little terminology thing. We call this over here a reducing end, and all of these are non-reducing ends. So where does that come from? It comes from the fact that in order to be a reducing sugar, you have to be able to linearize and expose a free aldehyde. Remember we talked about that earlier? And this is the only anomeric carbon in this whole molecule that is free and uncapped. You can imagine this little sugar here can open up and form a free aldehyde. So we call it a reducing end. All of the anomeric carbons for all other sugars, including these way out here at the tips, those are all locked and unable to linearize. Therefore, they stay cyclic and they can't do the reducing reaction. That's really just a terminology issue to help us understand structure. Okay, that's glycogen. Let's move on to the next page and talk about a couple others that are of interest here. This is cellulose. Cellulose is the most abundant biomolecule in the world because it makes up plants, trees, and shrubs, and leaves, and cotton, and all sorts of things. So plants are structurally held together by cellulose. So let's make a note of that. In this case, plant structure, wood, leaves, things like that, is held together by cellulose. Cellulose is also all glucose, but its linkage is different. And it's also linear. It's not branched. So let's go ahead and have you name this linkage here between these two glucose. Another good way to practice. Pause and let's see what you come up with. Okay, how do we name linkages? Well, I would always start by finding the anomeric carbons on whatever sugars are linked together. Then perhaps numbering them out. That makes it a lot easier. Now we can come in and say 1 to 4. That's a very common way to link them together. And next, we would say that in this case, the linkage has one anomeric alcohol buried in it. It's above, and on the same side is that flagpole carbon. That makes it a beta. So it's an interesting fact. Glucose is held in your body in the form of glycogen, which is alpha-1,4 linkages, and it's held in plants by beta-1,4 linkages. What's the difference? Why do they have this difference? Well, for one thing, cellulose, it undergoes a strange structural rearrangement in which every other glucose is going to flip upside down. It's hard for you to see that, but if we took this left glucose and flipped it upside down, it would look like this one over here. And when we do that, here's what happens. You end up having hydrogen bonds form between adjacent subunits. And these H bonds lend great strength and make this into more of a fiber than a food storage place or an energy storage. So these add strength and cause these fibers to make things like cotton and tree branches and leaves, stuff that's really tough. Lastly, we would say that we don't have enzymes to break down cellulose in our body. That's why we can't eat grass. That's why we can't eat tree leaves and things like that. But we do have enzymes to break down alpha-1,4 linkages, and that's why we can take out the glucose from our own glycogen. So these are common polysaccharides, and they're much more simple than, say, even lactose, which has different sugars in it. This is all glucose.